What do you think of when you think of the words gene therapy? Well, I'm sure it depends on what you have heard about it. A miracle cure, perhaps? Or a death treatment? Why did these two extremities surface? In 1990, 4-year-old Ashanti De Silva would have died if doctors did not use gene therapy to save her life. She was suffering from severe combined immunodeficiency, SCID. Because of gene therapy, she is now able to lead a normal life. However, in 1999, 18-year-old Jessie Gelsinger died in a gene therapy trial. The cause? The doctors who craved success for their trials accepted Jessie as a participant even though he was not medically fit. This was a huge tragedy. So we see that gene therapy can destroy yet save lives. Why? First, let's find out about how gene therapy works. We have millions of genes in our chromosomes. These genes are like the codes by which our body functions. The moment one of the codes is wrong, it could produce a genetic disease, like SCID. So what does gene therapy do? Plain and simple. Just insert an effective working gene in place of the mutated one. This then brings about two questions. Firstly, how can we insert the gene into our chromosomes? The best way currently is by a virus. A virus causes disease in our body by inserting their gene into our chromosomes, so they are well able to carry out the job. However, this causes the person to suffer the symptoms of the disease that the virus carries. No problem, scientists say. In the virus DNA, just remove the genes that cause disease, but leave the genes that infect the human cell. After which, we insert the treatment gene in, and the virus should not cause its disease. Does it really work? Perhaps. Till this day, no gene therapy case has had the virus regain its ability to cause its disease. The second question is, how can we ensure where the treatment gene will be inserted? The problem is, we don't know. A virus inserts its gene randomly at any point in our 46 chromosomes. This is the danger of gene therapy. In a gene therapy clinical trial in 2000, 5 boys were treated for SCID. After observing them for 2 years, doctors concluded that they were successful cases. 1 year later, however, 2 boys developed leukemia-like symptoms. What was the cause? What happened was this. The treatment gene, IL-2RG, was inserted near a cancer-causing gene, LMO2. LMO2 was activated to produce too much white blood cells, which is a leukemia-like symptom. The insertion of a gene into the chromosome that causes another problem is called insertional mutagenesis. So what have scientists done to solve this problem? In 2010, Zhou and his colleagues created a new self-inactivating lentivirus which does not activate LMO2 even when the treatment gene IL-2RG was inserted near it. They proved this in experiments done on mice and human bone marrow cells, showing that the problem of insertional mutagenesis could potentially be solved in the near future. As we have seen, gene therapy is no miracle cure, but it does prove as a potentially very good option for treating SCID besides bone marrow transplants.